Thank you very much for the introduction, and sorry I had to rush a little, as I'm legging it off to Cornwall as soon as this meeting is over for the weekend. I've got very little time to get there before dinner tonight, so I'm going to have to move along. So anyway, good to speak to you today. Media. I've forgotten this was about media, and I'm not really sure why I'm talking about quantum biology, but we'll plow on and see if we can find some sense related to media. So, quantum mechanics and biology. Does biology need quantum mechanics? Well, we'll see. First of all, how many of you are physicists? Put your hand up if you're a physicist. Ooh, quite a lot. Gosh. So most of you are physicists. Interesting. Okay, so maybe that gets me to Cornwall a bit quicker, actually, because that means I don't need to spend so much describing the difference between classical and quantum mechanics. But I'll point out some features that you don't necessarily always think about. Classical mechanics is the mechanics really of large numbers of particles. Newtonian physics, thermodynamics, etc., their laws depend on huge numbers of particles. Quantum mechanics depends on fewer particles. It's the physics, the mechanics that describes small numbers of particles. And it has all sorts of weird stuff that we're going to be hearing about. Things like the famous Schrodinger's cat and the double slit experiment, which demonstrate many of these features of quantum mechanics, which are features that are unfamiliar to us. We call them weird, but really they're not weird, because quantum mechanics is the fundamental basis of reality. What we see is weird. The fact that we don't see wave-particle duality, coherence, quantum tunneling, that's what we don't really understand. Completely, at least. Now, I'll just remind you, most of you seem to be physicists, so I'll remind you of this, one of the most famous experiments in physics, one that Richard Feynman claims all of quantum mechanics is contained in this experiment, which just illustrates the difference between particles and waves, and the peculiarity then of quantum mechanics. This is, of course, light going through the two-slit experiment. Generates two beams. Light comes as a single wave. It goes, or can be described as single waves, which pass through both slits to form two beams. And then those beams interfere with each other by the time they get to the screen. And that interference patterns where the peaks coincide of those interference patterns generates the bright patches, and the dark bands are where the peaks and troughs coincide, cancelling out the light. So that's what we're familiar with in terms of a wave phenomenon such as light. The waves in the sea will generate a similar kind of interference pattern. That's different from what we expect for particles, such as we see here, sand. Sand doesn't go through two places at once. It goes through either one slit or another slit and doesn't generate interference patterns. Now, if we... so this is really what I'm saying. Waves go through both slits at once. Particles go through one or the other. What if we deal with electrons? Now, electrons we generally or conventionally think of as particles. And if we send them through the two-slit experiment, but first of all, covering up one of the slits, so particles can't travel through this slit, then we get a sandpile, essentially, of electrons. If this is a screen sensitive to electrons, then we get essentially a sandpile of electrons, which indicates it's consistent, certainly, with particles. But what happens if we open up both slits? If we open up both slits, we get an interference pattern. So then there's a problem in understanding what's going on here, because we expect electrons to be particles to go through one slit or the other. And if they do that, they shouldn't generate an interference pattern, but they do. Now, what are the ways we can still try to understand that if it is particles? I mean, waves of the sea are made up of particles, atoms, or at least we think of them as atoms, and they still generate waves because the collection of all of the particles are moving together and generating the interference phenomenon. But the individual particles of water, we don't expect them to behave like a wave and have waves like the sea. So how do we distinguish between that and the kind of thing we see? Well, one of the things we can do is we can send particles one at a time through the double-slit experiment. We can send electrons with a gun that will pop them through one at a time, and then we would expect that any collective behavior will not be seen again. So we will go back to the sandpile kind of result. And when we do that, we send the electrons through one at a time, and they seem to behave like particles. They're hitting the screen as particles, generating little dots. But the weird part is when you increase the number of these and fire more and more of them. And as you fire more of them, you see, there's a bit of a pattern emerging. And more, you get an interference pattern. So even though you were firing electrons that seem to be leaving the electron gun as particles, arriving at the screen as particles, they still seem to be able to pass through both slits of the double-slit experiment as a wave. Now, that's puzzling, and you kind of think, well, surely an electron must go through one slit or the other slit. So let's find out which slit it goes through by putting a detector on one of the slits. And when we do that, here's our electron detector, we lose the interference pattern. And that seems weird. Why should the detection of an electron going through one or the other slit eliminate the interference pattern? Maybe it's the presence of the detector itself. It has some weird kind of gravitational effect maybe on the electrons going through the slit or something odd. So let's turn it off. And the interference pattern comes back. So this is really, how is it that this interference pattern, when we're not looking at it, it generates the interference pattern, but when we look at it, it doesn't. Something very weird and fundamental to quantum mechanics is going on here. And actually, it's interesting. So it seems to be that the particle is leaving the electrons are leaving the electron gun as a particle, but then they form some kind of wave that is able to go through both slits and appear as two waves that can interfere with themselves. But then when they arrive at the screen, then they arrive at the screen here as a particle. So somehow they start off as a particle, turn into a wave, and return to being a particle again. Now this is, of course, really weird. And yet when you put a detector on the system, they return to being particles all the way through the experiment. So what is going on? Now, to illustrate how weird this is, I'll point out an area here. This is a bright area of the screen when we have the detector present. So an area that's bright, but actually this is an area of the screen that is dark when the detector is switched off. Remember, when the detector is switched off, you get interference patterns. So it happens to be in a region of that interference pattern that is dark. And what we'll do is we'll put you there. And we'll imagine that you can see the electrons. So you are standing here and watching the electrons as coming through. Imagine them as light. And what do you see? Well, now with the detector on, you see both slits very clearly because the electrons are going through both slits. But you switch now the detector off, and you don't see any light coming through. Now, all that's happened is you've switched the detector off, which is detecting electrons coming through this slit. But somehow it's having a non-local effect on electrons coming through here. How can what you do here affect what goes on here? So that's one of the weirdest features of the double-slit experiment, and one that illustrates some of the key features of quantum mechanics. So as we say, what is happening here is electrons are both waves and particles at all times, but different experiments will reveal different aspects of them. So it demonstrates wave-particle duality. The other feature is that the electron wave going through both slits is coherent in the sense that what is happening at one slit will also affect what is going on at the other slit. The wave goes through, of a single electron goes through as a coherent particle through the system. And it can also be considered as a particle that's in a superposition of two places. It's in both places at once, in one slit and the other slit at the same time. That's my kind of very brief introduction to quantum mechanics and the weirdness of quantum mechanics. But there are many other features of quantum mechanics that I'll just very quickly skip over: quantum tunneling and entanglement. Quantum tunneling allows particles to travel through barriers that are classically impenetrable. Now, again, it's a weird feature of quantum mechanics, but it's responsible for the sun shining. There isn't time really to go into it, but basically, in a hydrogen fusion, hydrogen couldn't fuse if hydrogen nuclei couldn't tunnel, pass through an impenetrable barrier by quantum tunneling. So that's another feature of quantum mechanics, tunneling. It makes the sun shine. Another is entanglement, and again, this is one of probably the weirdest feature, where particles that are separated can still somehow communicate, entanglement. Now, all of these features have been implicated in quantum biology, which is why I'm mentioning them. And one of the ways I think that is useful to look at this is we can consider physical reality as consisting of different strata. At the highest strata is Newtonian physics. You know, the kind of things that cannonballs, planets, apples falling out of trees are described by Newtonian physics. Beneath that is the level of statistical mechanics, thermodynamics, the fluid laws, the gas laws, etc. And then beneath that is quantum mechanics. So those are the kind of three strata of reality. And one of the key things to point out again is that particles get more numerous as you move up. Quantum mechanics is revealed when you look at few particles. Now, in the previous slide, I drew the arrows this way, but of course, the reality is the causality is this way. Quantum mechanics is the bedrock of reality, and it causes all the rest. But at some stage, much of the weird stuff is filtered out. So you don't see the tunneling, the superposition, the coherence, the entanglement taking place at these levels. And this is how we get our classical laws. Through a process that isn't entirely clear, much of the weird stuff is filtered out from the reality that we see. Now, where is life in this? One of the things to look at first is the history of the road to quantum mechanics. As you know, went through Newtonian physics, Planck, Bohr, etc. At the same time, molecular biology, we have a road to molecular biology, and there was some interaction between molecular biology, or between biologies that was developing, say, Newtonian physics and the work of Descartes, René Descartes, who tried to build a materialistic biology. And later, some of the quantum pioneers became interested in genetics. One of them was Erwin Schrödinger, who of course is famous for his Schrödinger equation and Schrödinger's cat, etc. And he became interested in the process of heredity, particularly, and how it works. And basically, his argument, and I won't read out the quote, but his argument was that heredity is too perfect. When we inherit the genes from our parents, the accuracy is extremely good. An error 1 in, say, 100 million. And he said that to get that kind of level of accuracy out of classical mechanics, you have to have billions of particles involved. But genes are small. He worked out that genes were small. Too small to be accurate by classical mechanics. So he argued that the disorder must be somehow removed. And one of the ways of putting that is that at absolute zero, the temperature is, at absolute zero temperature, all disorder is removed. Now, obviously, life doesn't live at absolute zero. So the other way you get disorder removed is you deal with individual particles, or small numbers of particles, and then the laws are based on quantum mechanics. So he argued that life is rooted in quantum mechanics. Now, was he right? This is his proposal, really. This is the line you've just read. But he argued that genes behave like highly structured solids, something like a crystal that encodes information. An aperiodic crystal, he called it, whose rules were governed by quantum mechanics rather than classical mechanics. And it's this feature which provided the accuracy of heredity. Was he right? At the time he made that proposal, nobody knew what genes were made of, but of course a few decades later, the structure of DNA was worked out. And the remarkable feature of the genes in your cell is they are a single molecule. DNA is a single molecule. The color of your eyes, the shape of your nose, maybe your intelligence or propensity to disease, is determined by a single molecule that you inherited from your parents. One from each, of course. Now, there's nothing in the macroscopic world that has that sensitivity to single molecules. And if you were to ask a physicist, as there are many in the audience, what determines the dynamics, the properties of single molecules, well, you have to go to quantum mechanics. The code of DNA is actually determined by the position of protons. These are protons here between two DNA bases in a double helix. So the genetic code is a proton code. It's a code made up of the positions of protons. So clearly, genetic information is a quantum code. The fidelity of genetic information is written at the quantum level of particle position, proton position. So he was right. But, and it's a big but, by and large, as physicists say, for all practical purposes, you can forget about that. Geneticists mostly have. I'm a geneticist, really, and mostly we don't deal with quantum mechanics, hardly ever. So, for all practical purposes, heredity can be treated classically. And all of biology tends to be treated classically. This is an enzyme. I'm going to be talking a little bit about how they work in a moment. But the way that biologists, biochemists, like people here, usually understand how enzymes work is they deal with them as a ball and stick molecule, enzymes and substrates, classical objects that are behaving pretty much like Newton understood how cannonballs were moved in space. So by and large, biologists treat the subject of their work, the molecules like enzymes and DNA, classically. But despite that obvious mistake, they're not really classical objects, the approach has largely been successful. So what happened after Schrödinger's proposal? Well, pretty much it was forgotten. Nobody really paid much attention to it, and physics carried on regardless of biology, and biology pretty much carried on regardless of quantum mechanics. So does biology need quantum mechanics, and why? Why should it? So the classical view of biomolecules works pretty well, but are there phenomena in biology which cannot be accounted for within this classical picture? Well, yes, there now are, as we will come to now. So these are candidates for quantum effects in biology. The first five, there's some evidence for, or they're reasonably well established. Theoretically, these are very, very speculative, and I won't be talking about them at all today. So these are the leading candidates for quantum effects in biology, and I'll tell you why. We'll start off with magnetoreception in birds. So it's been studied often in the European robin. European robin is a migratory bird, different from our British robin, who, as everyone knows, who lives in England, stays here during the winter and famously hops around in the snow-covered trees. But the European robin migrates from northern Europe down to southern Europe or North Africa. And migration is, of course, not specific to birds. Lots of animals migrate. Turtles, whales, lots of fish migrate. So animal migration is well established, and to a certain extent we know how it works in some instances, but mostly we don't. Now, the Witskos, a couple who have studied the magnetic compass of the European robin. So it's been established by the Wiltschkows and several other scientists that many birds use a magnetic compass. They have a magnetic compass that can detect the magnetic field of the Earth. And, okay. And what they discovered, which was very interesting, is that it isn't a compass like a Boy Scout's compass that points north all the time. It's actually an inclination compass. And an inclination compass finds the nearest pole, but doesn't tell you which pole it is. And this is kind of how an inclination compass works. It actually measures the angle between the direction of the magnetic field, which at the poles is more or less vertical, and then nearer the equator becomes close to being horizontal. So what an inclination compass does is measures this angle and tells you where it's pointing. And anywhere away from the equator, this angle points towards a pole, any pole. Now, what the Wiltschkows discovered was that the robin's compass was an inclination compass. But no one had any idea how such a device would work. How do you make a compass? And how does it work in a biological organism? There's no evidence for any magnetite, any magnet inside the bird. And how does a field as weak as the Earth's magnetic field make enough of a difference to a bird to make it fly from Northern Europe to its summer home in North Africa very accurately? The Earth's magnetic field is extraordinarily weak. It shouldn't really make much difference to any biochemical reactions. But about the same time they were doing their work, another German, Klaus Schulten, in 1976, demonstrated certain chemical reactions involving free radicals were sensitive to magnetic fields. And the reason for that was that the unpaired electrons in free radicals, if they remain entangled, they become sensitive, or the reaction becomes sensitive to magnetic fields. The presence, absence, and angle of a magnetic field relative to the unpaired electrons orientation makes a difference to the final products of those reactions. So Schulten demonstrated that certain chemical reactions were sensitive to magnetic fields. And he went on, very interestingly, to propose that maybe these kind of chemical reactions may provide the avian compass. One of his students, Thorsten Ritz, went on to propose a model in which he proposed where are these free radicals coming from and how are they generated. Thurston proposed that the free radicals are generated by this protein cryptochrome, which is present in the eye of us and birds, and it can generate free radicals. So he proposed a radical pair mechanism for the chemical compass that depends on entanglement. For this to work, the free radicals that are generated in the proposed reaction have to remain quantum entangled. Now, that was a bold proposal because there wasn't really any evidence for it. And how do you maintain quantum entanglement in the retina of a bird? Seems unlikely. But he also predicted that if this was going on, the compass should be disrupted by magnetic fields, by oscillating magnetic fields, very rapidly oscillating magnetic fields, equivalent to the energy between the states. So actually, this is the more detail on the mechanism. So here's our unpaired electrons. There's the nuclei. Their spin states may be, when they become in a free radical, it may form a singlet in which the spin states are antiparallel, or triplets in which they're parallel. And in fact, what this system depends on is that they form a superposition between triplet and singlet states that is entangled. And he proposed that this was happening in the bird's retina. He made a prediction about that the energy difference between the singlet and triplet states should allow resonance with a high-frequency magnetic field. And he went on to demonstrate that birds' magnetic compass was indeed disrupted by megahertz oscillating magnetic fields. And it was very sensitive to the exact frequency. So it was a resonance effect. Something was resonating, and it was at least consistent with the mechanism that he proposed. So at least the entanglement model for the bird's compass is at least consistent with the experimental data that we have at the moment. There's still a lot to be discovered. There's still a lot of puzzles, but at least it's a good candidate for quantum biology. Next thing we're going to look at briefly is enzyme action. Now, this is an enzyme. This is a model of an enzyme. As I said, it's a kind of ball and stick model of what's going on. Enzymes are extraordinarily efficient. They can have factors of enhance. They're catalysts, of course, biological catalysts. But they can enhance chemical reactions by factors of up to 10 to the 20. So they're extraordinarily efficient, much more efficient than any inorganic catalyst. How do they do it is always a puzzle. And they almost certainly do it by a variety of mechanisms, including these here, but one of them seems to be quantum tunneling. This was first demonstrated in the 1970s with electrons. Electrons quantum tunnel inside your cells and are responsible for respiration, keeping you alive. Electrons are quantum tunneling between enzymes. So quantum tunneling is pretty well established in enzymes, particularly in electrons. It's certainly involved in respiration. Electrons tunnel between enzymes, and respiration wouldn't work if this didn't happen. More recently, this is showing what tunneling does. It's essentially a wave, a particle, when it's considered as a wave, can flow essentially through a barrier. But more recently, as well as electrons, proteins have been shown to be involved in tunneling in enzyme reactions. And this has been done by experiments in which they examine kinetic isotope effects. And basically, if tunneling is involved in a reaction, it will make a difference if the chemicals involved in the reaction which have hydrogen that is involved in the tunneling, if it's hydrogen, deuterium, or tritium. Now, hydrogen, deuterium, or tritium, obviously you're increasing the mass of the nuclei involved in the tunneling reactions. And if you increase the mass of a particle, then you slow down tunneling. Tunneling is very sensitive to mass. So if you measure an enzyme reaction in, say, ordinary hydrogen water and deuterated water and tritiated water, you can, by looking at these kinetic isotope effects, you can determine whether tunneling is involved. And this, for instance, is what Nigel Scruton at Manchester did for this enzyme, aromatic amine dehydrogenase. This is the reaction. It transfers a hydride group from this molecule to that molecule. This takes place in the active site of an enzyme, and it seems to involve proton tunneling, or certainly involves proton tunneling, because they get a kinetic isotope effect of 55. In other words, the reaction is slowed down by a factor of 55 when hydrogen is replaced with deuterium. And that can't be accounted for by any classical mechanism. Tunneling must be involved in this enzyme reaction. So, and similar experiments have been performed with many other enzymes and shown that hydrogen tunneling is involved in many reactions, and it seems to be at least partly responsible for some of the huge enhancement of enzyme activity that enzymes provide. And enzymes make everything in your cells. You have been made by enzymes. So if enzymes involve quantum tunneling is absolutely essential for life. Photosynthesis. Enzymes make stuff in your cells. The stuff in your cells has ultimately been made by photosynthesis in a plant or a microbe. Photosynthesis involves these chromophores, pigments, in reaction centers in leaves or in microbes that perform photosynthesis. They absorb a photon of light, and that photon of light is converted to an exciton. It moves an electron within one of these chromophores, which then oscillates between the electron and the hole. And that exciton then has to hop from one place to another until it reaches the reaction center. And in the reaction center, the energy of the exciton is converted into chemical energy, which then goes into fixing carbon dioxide, making the biomass that makes life possible on this planet. So that's all about capturing photon energy and transporting it rapidly to the reaction center. Now, recently, a number of groups used Fourier transform electronic spectroscopy to look at this transfer of energy of the exciton from its point of absorption to the reaction center. And they demonstrated that when they examined it by the process by this spectroscopy, they got what they call this quantum beating. Essentially, something was oscillating within the reaction center. And what is oscillating actually is the excitons seem to be traveling to the reaction center as a coherent wave rather than a particle. So this oscillating electron actually becomes a wave that then travels to the reaction center as a wave, and that allows it to more efficiently find where the reaction center is. If that exciton had to hop classically from one pigment molecule to another, it will mostly get lost and lose its energy before it reached the reaction center. But because it behaves as a wave, it can perform a quantum walk, essentially moving as a wave through the reaction center, through the photosynthetic center to reach the reaction center. So it means that this exciton is behaving as a wave at room temperature inside the leaves of the trees outside and in every other plant. And the really odd thing about it is that it's doing it for long periods of time. It has to remain coherent for this transport for relatively long periods of time. And how that is managed is not really clear. An interesting feature in the last few years, really, is that coherence in biology seems to be maintained by molecular vibrations. Now, we kind of think there's a lot of physicists here, so you know that coherence, normally you require to keep a system cool, keep it isolated, away from vibrations. This is why quantum coherence, entanglement, tunneling in biological systems is puzzling, because they're hot, messy, noisy systems where you wouldn't expect coherence to survive for very long. But it does. And the understanding now seems to be that actually biology has somehow worked out how to use vibrations, use molecular noise to maintain rather than destroy coherence. There's a lot of evidence for that. Now, the last one that we're going to talk about is DNA mutation. This is the one that first made me interested in quantum mechanics in biology. I'll just follow up really on what Schrödinger was proposing, that he proposed that genes were quantum mechanical entities. He proposed that mutations, where a gene is changed, are caused by some kind of quantum jump. Now, is this feasible? Well, when Watson and Crick looked at the structure of DNA, they pointed out that mutations may be caused by tautomization. And tautomization is when a base, which is part of the coding bases in DNA, moves from one place to another. The movement of a proton, essentially a quantum mechanical event. So, if tautomization is responsible for mutation, what happens is when this proton moves, the thymine, instead of pairing with its proper pair in DNA coding, which is adenine, it will pair with the incorrect base. And then will cause a mutation if the proton moves to this other position. But there are many causes of mutation. We know there are many classical causes of mutation. So it may be involved, but there are many things that cause mutation. This guy, Swedish physicist, he proposed a mechanism in which proton tunneling can cause these tautomers. So proton tunneling from one site to another, in which a TA pair in which this proton moves across from the thymine group to the adenine, and this proton moves, in order to maintain the charge, this proton moves in that direction. That can cause tautomerization, he proposed, and the same with another pair. And once it's moved, it can pair with the wrong base to cause a mutation. He proposed that in the 1970s. I got interested in it to see if it could account for a peculiar form of mutation called adaptive mutations, which were described in the late 1990s. I won't go into the detail of what adaptive mutations are because time is short, but they were hard to explain classically. So we proposed that adaptive mutations were caused by the load-in type of proton tunneling. Myself and Jim Al-Khalili, we proposed, wrote a paper back in 1999 proposing this. Essentially, what we were proposing was that I think it's probably not time to go into this explanation really, but essentially we proposed that quantum mechanics proton tunneling was involved in this phenomenon. It made sense, and if anyone's interested, you can read our paper. But it's hard to prove. And the problem with adaptive mutations was it became messy. But we wanted to go back recently and have a look at this with all of the recent interest in quantum biology. So one of the things that we're doing at the moment to investigate this is looking to see if mutation is perturbed by switching between hydrogen and deuterium. Same kind of experimental idea that we were looking at with the enzyme. If you replace hydrogen with deuterium in enzymes, you perturb enzyme activity. We want to ask the same question with mutation. If you replace hydrogen with deuterium in DNA, does it change the mutation rate? So we've done some preliminary experiments on this. These are colonies of E. coli. These are mutations here when they turn blue for reasons that we don't need to go into. And if we look at the rate of that mutation in ordinary water, in 50% deuterated water, in 100% deuterated water, that rate goes down. So that is only very preliminary evidence, and there are classical ways in which replacing hydrogen with deuterium can change how cells work. So again, it doesn't prove that quantum mechanics is involved. But it gets us a little bit closer, and we're currently trying to investigate this at the moment and find out more about this system. So, does biology need quantum mechanics? Yes. But it isn't clear how much yes. Certainly enzyme action, certainly photosynthesis, probably avian navigation, probably, I'd like to think, DNA mutation, but we'll see. So life does seem to be rooted in the quantum level. Certainly very fundamental aspects of life. Enzymes, they make everything. Photosynthesis makes all the biomass on the planet, is rooted in quantum mechanics. So biology does have this deep roots that go down to the quantum mechanical level, unlike the other stuff that we see, whose roots actually stay within the Newtonian mechanics or the statistical mechanics level of reality. But life is peculiar. Our argument goes, life is peculiar because it is a macroscopic phenomenon that portrays quantum mechanical dynamics at a macroscopic level. Some of the features of how our body works every day to keep us alive depend on these weird aspects of quantum mechanics, the weird stuff that is filtered out in most other macroscopic phenomena we see. But life somehow retains its connection right down to the quantum level. So that's what we're proposing anyway. And I think that's the last slide. So does biology need quantum mechanics? Yes. It isn't clear how much. How is the biggest question. Obviously, it'd be very interesting to work out exactly how biological systems maintain coherence, entanglement, tunneling at room temperature inside our bodies, inside the leaves of plants. And that could even have application in other aspects of technology. People who are trying to make quantum computers, for instance, making room temperature quantum computers that would work like a leaf does at room temperature. That would be great. So understanding how life manages to maintain coherence, superposition, entanglement, or whatever at room temperature could be very interesting even to non-biologists who might be interested in using this information. And as I said, one of the key interesting features that is appearing in the last few years is the interaction between vibration and quantum systems. That vibrations seem to be utilized by life in order to maintain coherence. And that's all I have to say. Thank you very much.